Bem-vindos ao seminário de hoje, que vai ser apresentado na área das ciências alimentares, engenharia alimentar. Vai ser apresentado pela Katarzyna, que vai ser apresentada pela Catarina Prista, que a convidou para este seminário. E por isso eu vou passar a palavra à Catarina. Thank you very much, Katarzyna, for your seminar, and welcome. E Catarina, podes avançar para fazer a apresentação.
Okay. Good afternoon, everyone. I just want to briefly thank to Katarzyna Polanowska, who works at the Poznan University of Life Sciences in the food department, and she's a teacher on food, on fermented food technology, and she will explain as a little bit of a part of her work, because it's more vast than this one, a part of her work on tempeh as a functional food. Thank you, Katarzyna, for being here, and the stage is yours.
Okay, thank you very much. Okay, so we will make some adjustments. Okay, let me take my mask off. It will be more comfortable. Okay, so Catarina looks me, and we can start. Okay, so Katarina already presented me. My name is Katarzyna Polanowska, or you can simply call me Catarina, like in Portuguese, it's similar name. So I am a teacher at Poznan University of Life Sciences, and also I work as a scientist there. Today, I will present you a little bit about myself, about my university, and also a little bit about the city where I come from. And then I will change topic to the main one, which is about tempeh as a functional food. And there I will give you some general information about tempeh as such. And then I will present you some results of my research. Okay, so about me. So I am like fully food technologist, because I made a bachelor and master degree in the field of food technology and nutrition at Poznan University of Life Sciences. And then I did doctoral studies also in the same field, in the department of the fermentation and biosynthesis. And I was working on tempeh fermentation to improve the quality, the nutritional quality of Faba bean. Meanwhile, when I was a master student, I had the chance to do the Erasmus in Castelo Branco. So I know a little bit more about Portugal already. And then during doctoral studies, I was able to do also several internships, scientific internships. The one of them was in France, in Montpellier, in CIRAD, where I was working with the molecular biology tools to identify the microbiome of the pulque, a Mexican beverage. And then several years later, I had the opportunity to do also some internships here in Lisbon and Nelnek. I was once here for 3 months. The other one, the other internship was 1 month, and I was working here with Professora Luisa Fozeira and with Rafał Łukasik. Currently, I am doing also the postgraduate course in project management at Wrocław University of Economics. And this is the most recent thing that I am going to do the internship in Glasgow in a biotechnological company producing mycoprotein, and I am supposed to develop products that will replace meat based on it. And I got this scholarship from EIT Food. Okay, sorry. Okay, and about the university where I come from, It's situated in Poznan city, in a very nice area, which is surrounded by parks and some lakes. So it's very nice to walk there from time to time if we have time. But for students, it's of course very nice. So as you can see on the pictures, we have some older buildings. The university is already more than 100 years old. So we have some older buildings from that time, which are quite representative. But also we have some newer buildings that come from the communist times. As you can see perfectly on these pictures, the style is not maybe so beautiful, but they are still very useful. And our university has 6 faculties. And recently we had some merging of the faculties, and now we have 6. So one of them is our Faculty of Food Science and Nutrition, but also we have faculties of Agronomy, Horticulture and Bioengineering, Faculty of Forestry and Wood Technology, Faculty of Veterinary and Medicine and Animal Science, Faculty of Environmental Engineering and Mechanical Engineering, and Faculty of Economics. And my faculty of Food Science and Nutrition, there we teach within main areas such as food technology. And within this area, we have several departments about meat processing and about foods of plant origin also, and fermentation and so on. We teach also within food quality and safety, food biotechnology. Food biotechnology plant is placed in this reddish building with the glasses. It's the newest one and quite modern. The main department, the main building is presented on the first photo, and also my department, my lab is situated there. Also, we have human nutrition and dietetics, which are quite strong recently because we have a very strong group within nutrigenomics, which is a very interesting subject. And city, where I come from, Poznan, as you can see on the map, it's quite far. This red points show you the distance. It's around 3,000 kilometers. And Poznan is located in the western Poland. Usually I explain it's as it is placed in the middle of the way between Warsaw, the capital of Poland, and Berlin. So we have 300 kilometers to Berlin as well as to Warsaw, more or less. So it's not a small city at all because it has 500,000 of inhabitants. And taking into consideration the whole agglomeration of Poznan, it's more than 1 million inhabitants. So it's quite huge. It's the 5th largest city in Poland. So of course, first is Warsaw, then we have Kraków, Gdańsk, Wrocław, and then there's Poznan. And there is a beautiful Barta River. It's not the biggest, but it goes through the middle of the city. And here on one of the photos, I show you how it can look during the winter. Yeah, quite interesting. Yeah, it's an academic city. Students like the city a lot. Every year we have more than 100,000 of students. And every year we have more and more students from abroad, because also there are a lot of universities, 25 in general, from which 8 are public. But there is an idea from several years to merge the main universities, but as far nothing happened about that. So on the photos I present you the main square and also the Oh, something happened. Can you see me and hear me? Oh. Okay, sorry, something happened to the computer. Okay, yeah. So during the Second World War, of course, the city was quite destroyed, and then it was renovated. Okay, Catarina showed me that everything is fine. Very good. And afterwards, everything was renovated, and the present view, it's a little bit different than it was in the past, but still it's very beautiful and worth to see it, because in fact, that's the region of Poland where the Poland was set up. So let me move to the main topic of today's meeting, which is Tempeh as functional food. So at the beginning, I will present you some general information about Tempeh. I think most of you who had classes maybe or know Catarina, Professor Catarina, so you know a lot about Tempeh, but maybe I will provide you some new information. And then I will move to the results that I got during my PhD thesis, some of them. So Tempeh. So it originates from Indonesia, from southeastern Asia. It's the indigenous food from there, and it's been known there from more than 300 years, and is considered as a staple source of protein. The main raw material used for the Tempeh production is soy. However, I had some conversation with the professor from Indonesia that is also working in Tempeh, and she was assuring me that soy was not the main ingredient in the past. Indonesian people used to use different grains, beans, or nuts for the Tempeh fermentation. And also, quite common raw material is okara. Okara is the waste that is obtained after the production of the soy beverage and or tofu, and it's even cheaper than soy. But soy, in fact, in Indonesia is cultivated not from such a long time. That's why she was saying that. Okay, the main strains used for the Tempeh fermentation are strains of Rhizopus, Rhizopus oligosporus, Rhizopus oryza, and Rhizopus solonifer. But among them, Rhizopus oligosporus gives the best sensory attributes. It's the smell and the taste is the best. And there's a description of soy tempeh in Codex Alimentarius, and there is a description of its sensorial features. So flavor should be meaty, mushroom-like and nutty, while odor should be fresh without any odor of ammonia. And we probably, those who have some experience in Tempeh fermentation, we know that this ammonia can appear, especially during fermentation in the higher temperature or too long, or depends also on the raw material that is used for the fermentation. And composition of Tempeh should be minimum 15% of protein content, maximum moisture of 65%, but in the Tempeh fermentation, moisture is very important, because if it is too much, then it's not going to work. It will be not pleasant at all. And a minimum lipid content of 7%, and maximum content of crude fiber is 2.5%. Here on the photo, I presented you the old style of preparing of Tempeh. So initially, Tempeh was wrapped, packed in the banana leaves or in the teak leaves, and the inoculation was also done that time. So these Rhizopus strains were on the leaves, and then the fermentation went on in these wrapped leaves. And now I will tell you some information more general about production, because the production varies a lot depending on the raw material. This scheme concerns raw soybeans. Of course, if we use okara or other kind of beans, the production will be different. And also there are different variations of the production of the soy tempeh. So this will be just more general, but I think that's the simplest scheme, and also shows gives a good results in terms of tempeh quality after fermentation. So we start with the raw soybeans. Then a very important step, which is soaking. Soaking is important for tempeh because in that stage, which depends up to 24 hours, enzymes have the ability to be activated and hydrolyze different compounds that are contained in soybean. Also, microorganisms can start work. They are also here activated, and for example, lactic acid bacteria produce lactic acid, which acidifies the soy, which is important for further fermentation, and it prevents from growing other different microorganisms on tempeh, just Rhizopus. And also some undesired compounds are extracted during that step. So it also gives important here some impact. In my lab, we developed a bit shorter method, because when we prepare tempeh, we usually don't have this patience for such long soaking. So we just boil the soy for 10 to 15 minutes, and then we make the hulling. So the next step is the hulling. Then we have washing and boiling. Boiling depends on the variety of soy, of course, because we need to obtain a proper hardness up to 45 minutes. Then we have drying, cooling, and inoculating. Inoculating can be done with the pure strain or with some commercial starter, which is usually some rice or some ground soybeans with the spores of Rhizopus, different strains of Rhizopus. Yeah, then we have packaging, and in the package, the fermentation is done. So we have then incubating, and here there are different information about fermentation, the time of the fermentation, because it can last, depends, from 20 or 18 hours even up to 8 days. And the main tempeh that we can buy usually in the shops in the Western world, let me say so, it's usually up to 2 days. It's something that we are able to accept the taste and so on. But in Indonesia, depending on the island, there are a lot of islands, they even eat tempeh old as 8 days. So you can imagine the taste is very strong. I haven't occasion to eat it, but I heard that it's for the very brave people. But yeah. Okay. So afterwards, it can be eaten immediately, of course, but also we can preserve it. So the preservation can be done by the pasteurizing or by freezing, also drying. But actually, personally, I haven't seen the dried tempeh. Yeah, and that is it. How it can be served, tempeh. So it's just up to your imagination. In fact, can be fried, which I think is the most common way to prepare tempeh. It's also the most tasty. The tempeh itself does not have very strong taste. It's quite plain, but with some meaty flavor, maybe nutty, depends. So the taste usually it obtains with the sauce that is given to it, or with the spices that are added to the dish. So it can be baked, can be steamed, can be grilled, can be boiled, and then added to various foods, even in hamburgers or in some dishes like goulash. I saw some recipes like that in the salad, and so on. So now about health beneficial properties, where they come from. Of course, the health beneficial properties, they come from the raw material that is used for the fermentation. If it is soy itself has quite high the nutritional value, very good amino acid score, and so on. But fermentation can help it to be better. So, for example, it causes the degradation of oligosaccharides like stachys. Stachys is not something that is very harmful for us. It's even considered as a prebiotic, but in fact, it's not very social ingredient compound, because can cause some flatulences and so on. So we are looking to remove it from the beans usually. The next is removal or degradation of some antinutrients like trypsin inhibitors or phytic acid. Also, during fermentation, we have the proteolysis of proteins, and some peptides are created, peptides with antithrombotic and antioxidative properties, which is considered very beneficial. And it's further hydrolyzed to amino acids, which are absorbed better in our digestive system. But also when we consider further, for example, frying or baking and so on, it takes part in the Maillard reaction. So in fact, they are not so available, but depends how we prepare it. Okay, next change that is occurring during fermentation is isoflavone glycosides, which are transformed to I-gluconase. Which show also better absorption in our digestive tract, and thus they can bind to estrogen receptors in our body and can have a strong pro-healthy effect, for example, in the postmenopausal women, but not only. Can also take part in the prevention of some cancers, and the obesity can also be used in the treatment of the, some lung cancer, and so on. You can also find information in the literature about vitamin B12 production during the Tempeh fermentation. Actually, Rhizopus is not able to produce this B12, but if we use the commercial starters, which contain different strains, not only from Rhizopus, like Klebsiella pneumonia or Citrobacter freundii, that are strains that are able to produce vitamin B12. So in this case, we also obtain some additional property of this tempeh. During fermentation, also iron from the 2 species is transformed into iron from the 3 species, which is also absorbed easier from our digestive tract. And also Rhizopus is able to produce vitamins from the group B. In my research, I was more focused on the Faba bean. So I wanted to do the research about Faba bean and the changes during Tempeh fermentation. And this was because there was very little information about changes during Tempeh fermentation of Faba bean. There were like, when I was starting it, there was like, I don't know if there were 3 or 4 publications like that. Most of them were about soy and also about other beans, but Faba was not so common. And why I was also interested in that, because Faba bean is a very important plant, especially for the countries from the Mediterranean region. It can fixate nitrogen, and it has very good nutritional value. And when compared to soy, we can see that it has very little fat content comparing to soy, which has more than 18%, and Faba has 1.5, and it has much more starch. So looking at this, we can suspect that the fermentation can go a little bit differently than in the case of soy, because of course the medium used for the fermentation determines the changes. So I thought that it would be very interesting to look at that. Moreover, Faba bean contain L-DOPA, L3,4-dihydroxyphenylalanine. And this compound is also quite important because it's a precursor of dopamine. It is converted upon the DOPA, the carboxylase activity in our blood or brain into dopamine. So this compound is used in the treatment of Parkinson's disease, which is usually obtained through the chemical pathway. Why it is used, this precursor, and not dopamine? Because dopamine itself can't cross the barrier blood-brain, and L-DOPA can. But for example, patients with Parkinson's disease, when they take L-DOPA, they have a lot of side effects because L-DOPA is transformed to dopamine in the blood. So it has a very great effect in the end. So they have to supplement it with carbidopa. So this is the enzyme, the DOPA decarboxylase inhibitor. So it can, the L-DOPA can reach the brain and be transferred to the dopamine in that place. As far L-DOPA is the best drug for the treatment of Parkinson's disease. There are some other treatments and ideas how to treat these people, but still L-DOPA is used in the like highest quantities. So there are some information in the literature, scientific literature, about the use of the other sources of L-DOPA for the treatment of Parkinson's disease, like Mucuna pruriens or Faba bean. This is very difficult research, in fact, because we might think that researchers are risking the health of these patients. So it's sometimes better to give them just a drug than to treat them with some plants. But nevertheless, these results were quite promising because these patients were eating the natural ingredients, and they showed less side effects than after getting synthetic drugs. Okay, and what's interesting about these plants, not only faba bean, but okay, faba bean and also mucuna pruriens, that this content of L-DOPA can be increased, and can be increased during the germination. During germination, the L-DOPA content increases itself, but when we use some stressors during germination, we can even increase it much more. So, for example, we can use microwave, UV, or some elicitors to stimulate the stress in these plants and to produce more and more L-DOPA. But what was the most interesting for me in that time was that information about the possibility to produce L-DOPA by using Rhizopus oligosporus by the Tempeh fermentation. And there are several researchers from more or less the same time from the one research group that were fermenting different plants in order to increase the content of L-DOPA in these plants. So when I was starting my PhD, I wrote a project for that, and I succeeded and I got the money. And then I tried to increase this content of L-DOPA in faba bean. Okay, so what else about faba bean? Like most of the, like all plants from the Fabaceae family, they contain some problematic compounds called antinutrients, like saponins, lectins, protease inhibitors, phytic acid condensed tannins, and in the case of faba bean is vicine and convicine. And about this vicine and convicine, there's very little information about the influence of fermentation on that compounds. That compounds are responsible for the causing the symptoms of the hereditary genetic disease called Fabism. So this disease is characterized by the deficiency of glucose 6-phosphate dehydrogenase located in the red blood cells and causing the hemolytic anemia. And actually not exactly vicine and convicine, but rather their aglyconies, divicine and isoramyl, which are created usually after eating faba beans. They are created upon the activity of bacteria in our digestive tract. So the other idea was to decrease the content of these compounds during Tempeh fermentation. Okay, the other thing within this aspect is that there are some varieties that contain low tannin and pyrimidine glycosides, but they are not so common yet. And in fact, they are very difficult to cultivate because they show worse resistance to the environmental factors. And also, it's quite difficult to maintain this variety is pure because bees can cross-pollinate these plants. I found some information in that time that, for example, lactic acid bacteria are able to degrade, to decrease the content of vicine and convicine in the faba bean. and also decrease the content of vicine, of the vicine and isoramine. So my question was if we can do the same with Tempeh fermentation. So objectives of my research were to see if we are able to increase the nutritional value of the Faba bean by decreasing some anti-nutrients or creating some new compounds that are beneficial to our health. Also, the other idea of my research was to compare this method of fermentation with some non-fermentation method that would also decrease the pyrimidine glycosidase content in the Faba bean. And at the very beginning, in fact, during my research, I had to develop the method for the L-DOPA detection because there is a lot of different, very different information in the literature saying, proposing different ways to extract it and then analyze it. So I had to develop my own, which was quite simple and fast. That was also very important because L-DOPA is a compound that degrades also very easily. How looked my fermentation process, how I did it. So firstly, I started from the fermentation of the whole ground Faba beans. And the fermentation lasted 6 days, was carried in 30 degrees, and the samples were taken every day. And this fermentation was done by 3 different strains of Rhizopus orygosporus. In these samples, I checked the sterol and fatty acid content, amino acids, proteins, vicine and convicine, yeah, and anti-radical activity also. Then I fermented only seeds whole. I did so because Faba bean contain a lot of tannins, but in fact not so much some free polyphenolic compounds, and I thought that this way might be easier to detect them. So the fermentation was done in the same way, and in the samples I checked the total polyphenol content, anti-radical activity, and also condensed tannins. And then to detect the degradation products of L-DOPA and vicine and convicine, which are also supposedly toxic, I decided to do the fermentation in the model conditions. So here I used a very simple media that was composed of starch, glycine and glucose. And I added to some samples the L-DOPA chemical standard, and also to the other samples extract from Faba beans cotyledons that represented the vicine and convicine. There was a lot of this compound. I didn't use the pure standards because the standard of vicine is very expensive, and of convicine even does not exist on the market. So I thought that would be the best way to do that. And the chosen strain for the fermentation was this choosing was based on the previous research that showed that this strain is capable to degrade these compounds in the higher level. So moving now to the results. In case of L-DOPA and vicine and convicine, as you can see, all 3 tested strains contributed to the decrease of these compounds. That the decrease was quite significant. For L-DOPA, the total degradation was already achieved in the 2nd day. So as you can see here, I did a lot of different repetitions of this fermentation and so on, and I never obtained results showing that the L-DOPA is increasing during Tempeh fermentation. And no one else in the further time, like further than the researchers that I presented you, repeated these results. So that was the first time that the opposite results were published actually. So unfortunately, the results were not as expected, but still it was results of the research. And in the case of vicine and convicine, which the results were more promising because this degradation was achieved. It was not so sudden. It was carried step by step, as you can see on this diagrams. And also, we detected the degradation products, divicine and isouramin, which contents also decreased during fermentation, which is also very important because this divicine and isoramil are responsible for the symptoms of favism. Okay, further, going to the results of fatty acids content during Tempeh fermentation. So what we can observe here? General content of the fatty acids decreased, definitely decreased during fermentation, but not all fatty acids were disappearing, were decreasing. Some of them were created. And one of them was palmitoleic acid, and also gamma-linolenic acid, which is actually, that belongs to the omega-6 family, but it shows the ability to decrease the inflammatory in our inflammation in our body. So this is quite interesting. But in general, when we compare the omega-6 and omega-3 ratio, it was worse after fermentation, but in fact the fat content in the Faba bean is not very high. But considering other products, we should take it into account. In case of sterol content, it was also very interesting because in case of Rhizopus oligosporus, main sterol is ergosterol. That's you can find information in the literature about ergosterol, but about other sterols, not necessary. And what surprised me a lot here was that the total sterol content increased in the samples in Faba bean, in fermented Faba bean, and also some of them were decreasing, like beta-cytosterol and avenosterols. But in the case of campesterol and stigmasterol, the content increased significantly. In case of proteins and amino acids, you can see on this figure 2, actually here, that there was a progressive degradation of different proteins that we were able to detect. And this was also reflected in the content of free amino acids. All of them almost increased. Also, some of them during the fermentation time initially increased, then decreased, because probably they were the source also for the fungi, the source of the nitrogen. You can observe on here on the slide that glutamic acid, the content of glutamic acid increased a lot, which is responsible for the umami taste, but also increased the content of gamma-aminobutyric acid, which was in fact not present at the beginning in the samples, and it was created during fermentation. This compound is a neurotransmitter which take part in very important processes in our body. And when supplemented externally, it can show some positive effects on our health. For example, it decreases the blood pressure, and also it alleviates from pain and some anxiety. In case of the computer is asking me for something. Sorry, I have some technical problems. Oh, now I don't have. Now it's fine. Now it's open, thank you. Okay, I managed. Okay, sorry. Okay, so now about total polyphenol content and condensed tannins and anti-radical activity. This result surprised me quite a lot because most of the papers describing the Tempeh fermentation and changes during Tempeh fermentation showed that these parameters usually not condensed tannins, but the rest were increasing in this in the other researches. And in my research, it was not. But in fact, the media, which was Faba bean, is different than, for example, soy or other beans. So it could drive the fermentation this way. Yes, also anti-radical activity decreased, which was also quite surprising. But I wanted to see more detailed results about polyphenolic compounds, and I ran my samples on HPLC to detect and to quantify and see if it is really true that everything during fermentation that all polyphenols are degrading. And these results are quite interesting because, as you can see in the table, some polyphenolic compounds like catechin or para-hydroxybenzoic acid, they decreased during fermentation, but the other ones, it depended because, for example, it was increasing. For example, until the third day, as in the case of camphorol or quercetin, and then it could stay in the same level or was decreasing later on. So here, the most interesting results I got from apigenin, which increased the most comparing to the other two compounds. For example, on the first day on this third strain presented in the table. And now I will tell you several words about the work that I did actually here in Lisbon, in Nelnek, because I thought that maybe not all people would like the taste of the fermented faba bean, and I wanted to develop maybe some method that would decrease the vicine content in the faba, but without applying fermentation. So we used this device that allowed us to use CO2 under high pressure. CO2 can act as a weak acid and can cause the hydrolysis of these compounds. So we thought that it might be a good idea. So we tested several parameters like the temperature, time, and CO2 pressure. in this device. We mixed the Faba bean with water, distilled water, in the ratio 1:10, and then it was subjected to this process. And after obtaining different results, we prepared these diagrams, and we could assess that at optimal conditions of 40.7 Celsius degrees, 5.1 minute, and application of 8.1 bar of CO2, we can extract vicine from the material up to 81%. So as you can see, the extraction time was very short, and the temperature was also not very high. So that are very mild conditions for the processing of bean, I think. And the results were better than the other results presented in the literature. We extracted the vicine on the higher level, but were worse than that ones obtained during Tempeh fermentation. So here still Tempeh fermentation is more beneficial, but this gives also a quite good result. And about extraction of L-DOPA, in fact, that was work that I was doing at the beginning of my whole journey during PhD. Because that was the main topic at the beginning. Then it had to change a little bit. So I was testing several here options. That was what was important for me, to choose the proper extraction solvent, which was composed of different concentrations of acetic acid. Also, I was testing extraction time, sonication, if it was needed or not, different times. Also solid to solvent mass to volume ratio and number of extraction steps, and some preconditioning prior to extraction. So we developed a method that needs homogenization. The concentration of acetic acid was 0.2%. The extraction time was 20 minutes, and the solid to solvent ratio was 1:25. And this method and that research was tested for several different varieties of broad bean. For broad bean, the big beans from the variety Major and Minor. So you can see different colors on this diagram. And in generally, the smaller beans had more L-DOPA content than the bigger ones. And the one that had the highest amount of L-DOPA in the beans was the variety, actually cultivar, called Fernando. Moving to the conclusions. So in general, the fermentation, Tempeh fermentation allowed us to create products with the higher content of some health beneficial compounds like gamma-aminobutyric acid, gamma-linolenic acid, stigmasterol, and campesterol. Also, it contributed to the degradation of pyrimidine glycosides and L-DOPA, and also their degradation products. And during fermentation, the protein was hydrolyzed to the peptides and free amino acids. Also, what we observed was decrease in the total amount of fatty acids, condensed tannins, phenolic compounds, and anti-radical activity. Also, due to the enzymatic activity of Rhizopus oligosporus, during this fermentation, some phenolic compounds were released. And the developed method, which did not use the fermentation, was more effective than many methods described in the scientific literature, but was less effective than the method using Tempeh fermentation. And the proposed method of L-DOPA extraction is a cheap, fast, and reliable alternative to the methods used so far, limiting the energy and cost consumption of the process and the amount of harmful reagents used. So thank you very much. And I'm waiting for your questions if you have any.
Thank you, Catarina, for your seminar. I have just one curiosity. I would like to know if tempeh is usually consumed in Poland or not. Or
tempeh, okay. I think it's similar as here. It's not a very common food like among older generations and so on, but among younger ones, which try to switch their diet into vegetarian, or they want to eat less meat.
Okay.
It's getting more and more common.
Okay. Okay. Thank you. Any other questions?
Hi, Catarina. Okay, let me see. Okay. I have one question, Catarina. I was curious about the reduction of L-DOPA in fava beans.
Okay.
And that was not what you expect. You expected that it increased, no?
Yes.
But I saw that you used the pure strains of Rhizopus, no?
Yes.
That, do you think that it may happen that like B12 vitamin or other compounds in the tempeh with Klebsiella that appears because Klebsiella probably is there, it will occur the same with the L-DOPA, or don't you think it may interfere? Because you didn't try the non-pure strains.
I used ragi. I used ragi, and
yes?
I had the same results. Yeah. At the beginning, that was what I was trying. And thank you very much for this question, because it might be like that. But actually, in these papers that I was at the beginning basing my work, they used some tempeh from the shop, but they were isolating the strains from the food. So in the end, they used some kind of the pure strain.
Okay.
Yeah, but also, you know, I had a problem with this method of extraction and the analysis of the L-DOPA because I was also basing this method of their method. And for example, they were, that was impossible to apply. And even the results showing very, very high amounts of L-DOPA in Faba bean, which was almost impossible. Especially they were using 95, I think, % of alcohol for the extraction. And when I calculated exactly everything, it was not possible to obtain such results because L-DOPA is very poorly soluble in the ethanol.
So,
yeah, so now I am very dubious about the results that they got that time, but yeah, that's the research.
Okay. And may I do another question?
Yes, please.
I was also curious about the ratio between the omega-6 and the omega-3 ratio that decreased, but you did quantify the amount of each one that you got, and it doesn't increase along the fermentation.
There was
besides the ratio, the amount of each one that you got.
Yeah, there was alpha linolenic acid and alpha lino. How is it in English? Lino. Sorry, it's one is from omega-6, the other one is from omega-3, and then it was like that this from omega-3 decreased a lot, but the other one from the gamma linolenic acid from omega-6 increased a lot, and that's why this ratio was unbalanced. Yes.
Thank you. Any...
Let's see in the chat, but I don't think we have questions in the chat. No, we don't have questions.
No, we don't have.
I think that we don't have more questions.
No. Okay. So maybe I would like to thank you to, I will end up, Manela. I would like to thank you to Catarina.
Thank you very much for giving me the opportunity to come, and that was a success to come. And this time, not so easy.
So, and thank you all for the attendance, and this will be was recorded, and so will be available for other people that wanted to see other time. Okay.
Okay.
Thank you.
Well, thank you very much again for inviting me here and giving this possibility. Thank you very much.
You're welcome.
Thank you, Catarina. Obrigada, Catarina, também.
Obrigada, eu.